So it's now my great honor to introduce our second speaker, Dr. Carol Chen. Dr. Chen did her PhD at the University of British Columbia with Matt Lawrence, studying H3K9 methylation, as well as H3 serine 10 phosphorylation in mouse embryonic stem cells. And I was lucky enough to work in the lab next to her during this time while I was working on my own PhD with Leanne Howe. And so I got to know Carol there. And from there, Carol then moved to Montreal for her postdoc with Nada Jabido, where she did really tremendous work on H3.3 point mutants in neurodegenerative disorders, as well as glioblastomas. Her CV is just a tour de force of fantastic papers. And then I'm very excited for her, as Carol has just begun her lab at BC Cancer's Terry Fox Labs at the University of British Columbia back in Vancouver. She's been the recipient of numerous awards, including a prestigious Young Investigator Award from Alex Lemonade Stand Foundation. And for people in the lab looking for technician, graduate student, or postdoc positions, she is hiring, and I highly recommend reaching out and applying to her lab. So with that, let me finish by saying I'm very excited for her talk today on germline histone H3.3 mutations in neurodegenerative disorders. So please join me in giving a warm, albeit virtual, welcome to today's second speaker, Dr. Carol Chen.
Thank you so much, Ben. That was an amazing introduction, and hopefully I live up to it. And slides are okay. So far, so good.
They look good.
I see thumbs up. Okay. Thank you to Ben and the rest of the organizing committee for this huge effort in bringing the community together. It's such a pleasure to be giving my first talk as a PI to all of you. And today I'll be talking about our story that was recently published in Cell last month, which is on the germline histone 3.3 mutation. So this slide really does not need to be introduced to this audience. You all know that our DNA is wrapped around histone proteins, and that both the DNA and histones are densely modified with covalent modifications. And these modifications serve to regulate transcription. And it's actually quite critical for processes in normal development. So it's been famously described by Conrad Waddington that these epigenetic mark forms a landscape that determines whether a cell can commit to a progenitor fate and stays locked into terminal differentiation. And in cancer, this concept also applies. So the underlying chromatin context may determine whether a particular cell type is actually amenable to transformation or disease versus another cell type that is would lead to a benign condition. So this is something we explore in disease in the brain. And my particular interest is this really interesting convergence between germline neurodevelopmental syndromes as well as brain cancer. So we've known for a while that germline neurodevelopmental syndromes can be caused by mutations affecting canonical epigenetic modifications such as DNA methylation, H3K36 methylation, and H3K27 methylation. And conversely, it's been more recently appreciated that in childhood brain cancer, particularly in high-grade glioma, the same epigenetic mechanisms are being perturbed, suggesting that this tissue, the brain, is particularly sensitive to perturbation of these pathways. So today I'll be focusing on histone 3.3 G34 mutations, which were originally described in glioma, but today I'm going to talk about our new finding in germline syndromes. So the G34 mutations are quite interesting in that they occur exclusively on histone 3.3, which is a non-canonical histone 3 variant that is deposited independent of the cell cycle. And these G34 mutations that occur in cancer happen in a few different flavors, so 3 in particular. So within brain cancer, we see arginine and valine substitutions, and altogether they encompass about 30% of hemispheric glioma within pediatric patients. Now, the third flavor, which is the tryptophan, G34W, is found almost exclusively in a form of bone cancer called giant cell tumor of the bone. And up until very recently has never been seen in central nervous system cancers. So there is this remarkable tissue specificity to how these individual mutations are found. So that is something that we'll try to answer today. And as the data I'll show you. Okay, so another piece of the puzzle came to us very recently. Our collaborator, Dr. Elizabeth Bosch, CHOP, has identified rare patients that carry germline mutations in Histone 3.3. This is remarkable considering that Histone 3.3 sequence is completely unchanged from Drosophila to humans. So having a germline mutation is quite shocking to us at the time. So unlike the mutations that we see in cancer, the germline mutations kind of scatter all across the amino acid stretch of 3.3. They don't pile up like the hotspot mutations we see in cancer. Nevertheless, we were able to identify two patients, one with a G34R mutation, another one with G34V mutation. To remind you, those are the ones that we found in brain cancer as well. And neither patient had any sign of malignancy, but they did show signs of global developmental delay. They were born with short stature, even compared to the rest of the 3.3 germline cohort. They also were underweight, and they showed signs of microcephaly. Both also showed worsening clinical symptoms that would be suggestive of progressive neurodegeneration. And what we think is happening is that the brain is particularly sensitive to mutation of Histone 3.3 because in postmitotic neurons, because of its deposition pattern, 3.3 can actually make up over 90% of the histone pool in this cell type. So now to investigate how G34 mutations, germline G34 mutations were affecting global development, Seema Kazai in Nada's lab generated direct knock-in transgenic mouse in the germline. So she utilized a CRISPR direct knock-in strategy that targeted the endogenous H3.3 encoding gene. And we injected the Cas9 as well as guides directly into 2 to 4 cell embryos to generate mosaic founders. And this step was actually quite crucial in getting viable animals that were able to transmit. These mosaic founders were then back-crossed into 2 different strains, and then we were able to obtain true founders that carried germline mutations in G34R, V, W, and heterozygous indel as control. And to our very surprise, each of the G34 mutations show completely distinct phenotype as the mouse age. So G34R essentially had fully penetrant neurological deficits exhibited by signs of ataxia, such as this response called hindlimb clasping. They also had abnormal walking patterns, abnormal gait. In contrast, the W mutation, which is found in bone cancer, did not show any overt signs of neurological defect, but in fact, these mice showed 100% penetrance for mesenchymal-related disorders. They actually died prematurely due to obstructed bladders. They had morbid obesity. And the G34V, on the other hand, showed an intermediate phenotype between the R and the W. And this really echoed the sentiment of the late Dave Alice. He stipulated that every amino acid matters. And in fact, within oncohistone, which amino acid you change it to really does make a difference. So we decided to focus on the G34R mutation because we were a brain lab and we wanted to know what the R mutation was doing on the brain. So we looked specifically at this hind limb clasping response because it was an easy phenotype to assess in a lot of mice, and we can easily score it for severity. So in a normal mice that did not have ataxia, when you pick up the mouse by its tail, it should splay out its limb. However, in an ataxic mouse, it will start clasping its hind limb and eventually will progress to a severe form where they start clasping their forelimb as well as both sets of limbs together to form a bat-like posture. So what we can see is that as the G34R animals age and reach adulthood, this response became more and more severe, again, showing this progressive nature of this phenotype. And in contrast, G34B and G34W did not show this. And I apologize for the scratching of the dog in the background. In terms of other motor tests, G34R and V also showed similar deficiency in terms of how much they traveled or how fast they were, whereas W were mostly normal to wild type. So morphologically, when we looked at the brain of the G34 animals, the R was also very striking in that the brain weights were much smaller than the other G34 mutants and in comparison to the wild type. This is exemplified here in the picture. And also when we perform histology on the cortex, we could see that there was the thinning of certain cortex layers, including these deeper layer 5, layer 6 of the cortex. And when we stain for motor neurons in the cerebellum, so these are actually responsible for walking and that hind limb clasping response, we can actually see that in newborn mice, the neurons are there. They're actually, they don't look very different from wild type. But as the animals reach adulthood, they seem to deteriorate, suggestive of neuron degeneration rather than a failure to specify neurons. And to further examine which lineages in the brain were affected by this mutation, we did a bulk deconvolution of the transcriptome signature from the cortex and compared the G34 mutants to the wild type. So the red cells are all the cell types that are over-enriched in the mutants, and the blue are the cells that are depleted. And what we can see right away was that the G34 animals had much more infiltration of immune cell types, the microglia, as well as reactive astrocytes, and were concurrently depleted of neurons. And when we look further into the signature, we can see that the microglia that were present in G34R brain was actually showing a signature that is normally found in neurodegenerative brain and is actually associated with disease rather than overabundance of normal microglia. And furthermore, the neurons, we could actually see that the deeper layers of layer 5 and 6 neurons are more strongly affected than the superficial layer, again, echoing what we saw in histology. We then confirmed all of this deconvolution using immunofluorescence or immunohistochemistry. We can see the infiltration of GFAP positive astrocytes really in the deeper layers of the cortex in the G34R animals. This was also seen in the microglia that are IBA1 positive. And concurrently, when we stain for layer 5 neurons with the OX6, we can see that they were depleted. Okay, so the phenotype was really robust. However, we did question why this was unique to G34R and not found in G34W. So what we knew about the G34 mutation from literature and from our previous work is that this particular histone mutation actually acts in cis, as in it impedes only the adjacent lysine 36 methylation, and this effect is only on the mutant histone. So it does this because K36 methyltransferases, such as SETD2, recognizes the K36 residue by threading H3 through a very narrow channel. And through this very narrow channel, G34 is actually needs to fit into this tunnel. And you can imagine substituting glycine, which is the smallest amino acid, to any of the bulkier amino acids, such as arginine or tryptophan, would impede this entry, and therefore SETD2 cannot methylate G34 mutant histones. So we decided to investigate this K36 methylation in mutant histone a little bit further. And we utilize histone mass spectrometry through our collaboration with Ben Garcia. And we looked specifically at cells with G34R versus G34V versus G34W. And this is the difference to wild type is plotted on a log scale with all of the bars under being depleted. And what you can hopefully appreciate is that G34R uniquely showed very strong depletion of K36 tri and K36 dimethylation. And this was quite exciting because K36 dimethylation recently, through the work of Chao Liu and Matthew Lawrence, has been shown to mediate de novo DNA methylation machinery, namely by recruiting DMT3A through the PWWP domain. It's actually necessary for many forms of de novo methylation during development. And we wanted to confirm whether G34R mutants actually impaired the interaction with DMT3A. So to do this, we performed bio-ID with our collaborators in Toronto, Eric Campos and Anne-Claude Gingras. And we saw that G34R mutant histones actually did completely abolish interactions with DMT3A and a bunch of other PWWP containing reader proteins. And this was not seen to the same extent for V or W. Now, why 3A, DMT3A may be critical in explaining this phenotype is because it's actually very well known that DMT3A is really important during postnatal maturation of the brain. So the mRNA and the protein is transiently expressed in a huge spike shortly after birth. So this is time point in the mouse. And this lasts about 2-3 weeks. And it's during this transient spike of expression that DMT3A deposit a specialized form of DNA methylation called CH methylation. So that is any residue that is not a G. And this mark is thought to be repressive uniquely in neurons. And the accumulation of this mark also coincides with synaptogenesis. And we were especially excited because reading from literature, we know that DMT3A knockouts exhibited or phenocopied many aspects of our G34R mouse. They had increased gliosis. They also showed hind limb clasping, which, as you all know, we saw in our mouse as well. So we figured that this may explain why G34R animals may show this impaired brain function. So to prove this, we decided to do epigenomic profiling in early postnatal G34R mice and wild type. And we first looked at K36 dimethylation. So K36 dimethylation is a very broad, pervasive mark. It marks about 80% of the genome in these broad domains. And in fact, what we saw in G34R is that the K36 dimethylation broad domains begin to contract. We actually start to see loss of K36 dimethylation away from in the intergenic regions, and instead it was accumulating at active gene bodies. And this pattern was followed by DMT3A binding, in that the binding patterns was shifted from intergenic regions into gene bodies. And when we performed whole genome bisulfite, we can see that CH methylation very much followed these changes as well, with loss of CH methylation being much stronger than the change in CG methylation. So altogether, in the genome, there was about 40% of genomic regions that showed significant loss of CH methylation, whereas the trend was still there with CG methylation, but it wasn't as dramatic. And while most of those changes occurred in intergenic region, there was one class of gene that showed preferential loss of CH methylation much more than any other class of genes. And there were these large clusters of innate immunity genes. So shown here, showing you the interferon locus, the MHC receptor locus, as well as the complement locus, and all of which showed this kind of broad loss of CH methylation that expands upon the whole cluster. So this was quite interesting, and we wanted to know whether this loss of CH methylation, a presumed repressive modification, would lead to transcriptional upregulation of these immune genes. So to test this, we intersected transcriptome data with our DNA methylation data, and that's plotted here on 2D scatter plots. And what we saw was that our immune genes, which are red, were concurrently demethylated as well as strongly upregulated in the brains of G34R. Conversely, neuronal genes, which typically are unmethylated, actually are gaining CG methylation, which may explain why they're being downregulated in the brain of G34R. So just go back. So what we saw really prominently in the transcription data was that the entire complement cascade was actually being represented in the epigenetic and transcriptomic signature. So that included complement 1 to C3 to C4. And that caught our attention because complements actually are very critical in postnatal maturation of the neuronal circuit. So neurogenesis in mammals actually occur in the fetal stages in utero. So neurons are born unspecified, and they're born in far excess of what they are in the adult brain. And shortly after birth, they're actually pruned away, and the excess neurons are removed and the synaptic connections are refined such that the resulting neuron circuit can mature. And mediating this process is actually the transient expression of complements in newborn neurons, which then recruits activated microglia to come to perform phagocytosis and to do refining of synaptic connections. It's famously been shown by Beth Stevens that complement expression happens in the first week of postnatal life in the mouse. And just to remind you, that's the first week. And then at P14, DMT3A is then turned on. So we hypothesized that DMT3A may be transiently expressed in the postnatal brain, in fact, to lay down the CH methylation and partially to silence these immune clusters from being persistently activated, and therefore you can stop synaptic pruning during development. And what may be happening in our G34R mice is that this halting of complement expression is not occurring. So to look at this, we perform some staining in the G34R brain, looking particularly at neurons and not immune genes. So we utilize neuronal markers, which are in green, and we stain for complements, which should be silenced in adult brain. And we found that in wild type, there was essentially no signal. But in the G34R animals, in fact, our neurons, their neurons were being tagged with complement proteins. And this also could be explaining why there's abundance of microglia, which have IGA1 positive. And indeed, when we co-stain for neuronal synaptic markers and microglia, we can see that a bunch of the microglia actually showed synaptic fragments within their cell body, suggesting that there is synaptic pruning occurring in the G34R animals in the adult. So we have a model for what's happening, and I'll start with the wild type. So we think that at birth, there is these chromatin modifications, namely K36 dimethylation, that is bookmarking regions of the genome, particularly the intergenic regions and innate immunity genes, that then allows for DMT3A, when it's turned on, to be recruited to those sites to lay down CH methylation. And this accumulation of CH methylation is what leads to stable silencing of these loci in adulthood. And what happens in the G34R animals is that this reduction of K36 dimethylation leads to an alteration of DMT3A recruitment, such that it's now leaving from canonical sites, so sites that should be methylating, such as the complement genes, and instead is inappropriately sitting on unmethylated CpG islands. And as a result, we have sustained activation of complements and in turn, inappropriate neuronal repression. So with that, I will close and thank everybody at McGill, my previous postdoc supervisor, Nada Jabato. So all of the great things that Ben has said about me, it's because Nada gave me the opportunity and really allowed me to lead. I'd like to acknowledge my co-first authors, Seema and Gusto, who have done so much mouse work and such beautiful, careful characterization of those mutants. And of course, our other collaborators, including Claudia Kleiman and Livia Garcia. And as Ben said, I just started my lab about 2 weeks ago, and we're located in beautiful Vancouver, BC, and I'm hiring at all levels. So please, if you want to live here and pursue studies of epigenetics and cancer and developmental syndromes, please shoot me an email or a DM on Twitter. And with that, I'll close and just thank you for your attention.
Thank you, Carol. That was really fantastic. I'll just note that we're all very honored that your first talk as a PI was for the fragile nucleosome. Yes, so I have a bunch of questions. There's one coming in. I'll just ask like a kind of a general question about kind of these H3.3 mutations and how they're having such a specific phenotype in the brain. I have vague memories of like in post-mitotic neurons, they start to accumulate H3.3 over the other variants. And so do you think part of why you get these kind of neuronal phenotypes is because H3.3 starts becoming a much bigger proportion of the histone H3 in the cell?
Yeah, that's definitely our theory. So furthermore, it may explain why neurons and not, let's say, oligodendrocytes or astrocytes, the glial cell types. So the glial cell types are thought to divide. So because they divide, you would presume that they incorporate canonical histones such as 3.1, 3.2. But in neurons, once they've stopped dividing, there's transcriptional activity. 3.3 eventually gets replaced into all of the genomic regions. So yes, we have never done the test where we introduced the G34 mutation into canonical histones, but I imagine that's something on Nada's list.
It's a much harder one to introduce though, with the number of
the 14 copies. Yeah, it's
okay, so we got a question from Alan Underhill asking if you can speculate on the mesenchymal phenotype with G34W.
That's a harder one to answer. I have to say that's kind of the next study. So we have a bunch of very interesting data on W that we were kind of hoping the reviewer would ask us, but they never did. So we didn't make it to the paper. So the W actually has, instead of hypo DNA methylation, it actually has hyper DNA methylation. And we do not know why currently, whether that is due to perhaps increased K36 methylation. There's some reports that the W mutation may be affecting demethylase activity. So that's to say I don't have an answer, but it's something we're very interested in.
And for that where you've got kind of like the cell types that those are popping up in, are those post-mitotic cell types as well, or is this something completely different?
So the bladder is mostly a sac. But we don't think it's the actual bladder. We think it's the muscle that is actually controlling the bladder. So we think this is a muscle phenotype rather than, let's say, epithelial phenotype. And you can imagine those muscles don't divide after a certain time. So we don't really know is the question or the answer really. Like I think the G34 mutation the R mutation makes sense because 3A is so critical for the brain. You can imagine that W is maybe affecting another chromatin modifier, but it's very critical for that particular muscle. We just haven't been able to identify it.
And then we have a question from Jason Fan who says, hi, very nice talk. I was wondering if you can speculate why the G34W mutation doesn't impact K36 methylation.
Yeah, so great question. I think it may have to do something with the dimethyltransferases. So if you look very closely at that SETD2 crystallography structure, the NSDs and ASH1L actually have slightly different amino acids in the positions that are contacting or that are around that channel. So something that we're working with Albert Burgess and some structural biologists is actually modeling each of these mutations separately to see why. But with respect to SETD2, I think or Gazani has actually shown, and Peter Lewis has shown that W is also impairing K36 trimethylation, but I don't know why in our mass spec we were not seeing a huge difference.
Great. And then one of our organizers, Matteo, has a question, so I'll let him ask you that.
Yeah, fantastic talk, absolutely. It was just a small curiosity because you mentioned the possibility of the bladder phenotype being muscle derived. So that is basically smooth muscle cells, which is also what is regulating, vascular size. So do you have any vascular phenotype that you have noticed in these ones? Because if you have a weakened, vasculature wall, that might also be.
Yeah, we definitely have hints that there is a muscle phenotype present in all G34 animals. They have dystocia, the females. So the dystocia is, essentially a smooth muscle phenotype. So I think there will be a defect if we look for it. They're just not dying of those vascular defects. So we've been focusing on the really fully penetrant phenotype. I think in the bladder phenotype, for example, is a male specific, or at least it affects males much more than females because of their anatomy, males are much more sensitive to bladder obstruction than females. So yeah, I think there's lots of interesting work to be done. And one thing that's actually has come to our attention recently is that Rett syndrome patients and the Rett mouse, the MECP2 mouse, actually also have urological disorders. So we don't know how much of this is neuronal muscle crosstalk or just a muscle phenotype. But yeah, very exciting. gonna work on that.
Very cool, thanks.
Thanks.
And Carol, for the phenotypes that you see kind of working through the K36 methylation, like you have reduced K36 methylation, have you thought about the demethylase and like if perturbing the demethylases could suppress some of the phenotypes?
Yes. So I think there was lots to go forward on these mouse models. So one of the question, first of all, was if we cross the G34 mutants to, let's say, complement deficient mice, are we able to rescue the neurological deficit? So one can imagine you can cross these to some of the chromatin modifier loss of function mutations to see if they can mediate this rescue. I think the trouble with the demethylase is that they often demethylate other residues as well. So like, you know, if it will hit K9 and K36. So I think we need to be really careful about which one we target.
Yeah. Awesome. Well, we're getting up to 1, so let's wrap up there. If you have further questions for either Carol or Stephen, I'm sure they would love for you to email them or message them on Twitter. And but let me just finish by saying thank you to both our speakers for a wonderful session. And thank you all for attending, and we hope to see you again in 2 weeks.